So, Antonio, you can.
Okay, thank you, Mithun. Welcome everybody to the latest installment of our series of colloquia, which are organized by IIT Bombay, and we as ICTP, let's say, collaborate in this endeavor. So we have had an excellent list of very distinguished speakers so far, and today is no exception. So we are very happy to have Anna with us today, and I will leave the floor to Mithun for further introduction.
Thank you, Antonio. So it's a great pleasure to welcome Professor Anna Erzberger from EMBL Heidelberg. Anna did her PhD from the Max Planck Institute for the Physics of Complex Systems in Dresden, where she studied using hydrodynamic theories, cortical actin-myosin flows in cells affect cell migration. She then did her postdoc work at Rockefeller University, New York, where she studied what are the physical principles associated with the developmental process of mechanosensory epithelia. She's been a group leader at EMBL since 2020, and her group works broadly in the area of biological physics on subjects related to active matter, non-equilibrium physics, morphogenesis, and nuclear mechanics. So today she will be talking on surface dynamics in living systems, and it's a great pleasure to have you, Anna. Before I hand over to Anna, I just want to say, so if you have questions in between, please raise your hands, and then one of us will ask you to unmute, and you can then unmute and ask the questions. All right.
Thank you very much for the introduction, and thank you so much for the invitation. I'm very, very excited to present the colloquium today. So as was said, we are a theoretical physics group at the Biology Institute, where we collaborate with experimental colleagues, and we use cellular and multicellular systems as paradigms to study how complex systems self-organize, and the focus for us is the role of geometry. So geometry that can be a property of the boundary of the system or a property of the domain itself on which patterning takes place, such as curvature. And biology, of course, is full of really interesting complex geometries that change. So they are dynamic, they grow in time, or they deform under self-generated stresses. And something else that is important is that in biology, a lot has happened in terms of the technologies that enable the imaging of biological shape dynamics. So I will discuss a few examples today. And basically, I'm very, very happy about the interruption. So if you have any questions, just immediately ask. All right. So in my view, a very fundamental aspect of spatial organization in biological systems is the interaction between structures that can be approximated as two-dimensional surfaces and the three-dimensional bulk that surrounds them. And many patterning processes and structure formation rely on such surface bulk couplings. Just to start with a bit of an overview and a few interesting examples, on the left-hand side, you can see my mouse, right?
Yes, we can see.
If not, thank you. Sorry. Yeah, so this is a very famous reaction diffusion system that controls patterns on the surface of bacteria. And in this very nice paper, the authors studied. So this patterning relies on exchange of molecules between the bulk and the surface. And here the authors have studied how the geometry of the bulk, the height of the domain, is a control parameter for different types of patterns that show up in the system. The middle example is a minimal model of a cell that undergoes spontaneous symmetry breaking and generates active flows, hydrodynamic flows at its surface. And these surface flows entrain the passive fluid in the bulk, and then we get these vortex structures on the inside. The last example is a very quick preview of work that is ongoing in my group. I will not talk about it today. Here we are interested in boundary-induced polar alignment and how the geometry, the curvature of the boundary affects the defects that form at the center. In biological physics in general, there has been a rich tradition, I would say, of using the analogy between bubbles and foams and cells and tissues. And there are many works and many modeling frameworks that rely on this idea. And basically what it comes down to is that the thermodynamic concept of surface tension, which can be defined as an energy cost per unit area, which has been developed in the context of things like a lipid layer at the surface of a bubble, that it can be generalized also to biological surfaces, which have, of course, much more complex microscopic structures. So this here is a schematic from a recent review, and I'm just putting it here to show you what a terrible mess it is actually. This is the surface of a cell. We have cytoskeletal filaments surrounded by regulatory proteins, active motor proteins that can generate stresses. They convert energy from metabolic sources into work. We have coupling to the plasma membrane, which has its own biophysical properties, bending, rigidity, and so on. So it is really, really complicated. But in many contexts, we can make the argument that on length scales and time scales that are relevant for cellular function, let's say, so we are talking about tens of micrometers and minutes to hours of time scale, we can neglect many of the microscopic dynamics that happen at this level, and thermodynamic coarse-grained concepts such as surface tension become relevant. And indeed, we can use it to very accurately predict, of course, so bubbles, we can very, very precisely predict the shapes, equilibrium shapes, given boundary conditions. But this is true also for some biological contexts in which some of these arguments survive. At the same time, active processes at these microscopic scales give rise to large-scale properties as well. Most importantly, we can have gradients in components of the tension, and these gradients can do work on the contact lines, and we can have self-propelled movement of droplets. And in the end, I will also talk about an example where we have two coupled droplets, and then we get rotations of the contact line in between them. But I will start with, so this is a bit an overview slide as well as an explanation of concepts. I will start with some examples where we can stay with very simple equilibrium arguments and then slowly move towards the more interesting non-equilibrium cases. So biological lumina, I think, are a very, very nice example of biological structures that can be viewed as general objects with an effective surface tension and subject to pressure, hydrostatic and osmotic pressure from the inside. These processes are regulated by biological processes, but at larger scales, we can view we can view these objects in analogy with bubbles or droplets. And there has been a lot of really nice work in recent years that is exploiting this analogy. Here I am highlighting a study where the authors have looked at the coarsening of microlumina, so these very small fluid field spaces inside the mouse embryo. They look at how these coarsen, and they find that the dynamics resembles the coarsening of phase-separated droplets. And similarly, we can study the initial formation of fluid field cavities in analogy with the nucleation of a liquid phase. And in this study here, the authors have actually started from considering various cellular processes that contribute to lumen formation, including osmotic pumping and the cytoskeletal active tensions at the surface of the cells that are adjacent to the fluid. And they end up with an equation that looks completely like classical nucleation theory. So we have this dynamical equation for the radius of the lumen, where we have a bulk term that acts to inflate the lumen and a limiting surface term here that scales inversely with the radius. And we can immediately from this equation get out the critical radius below which, so lumina which are smaller are expected to shrink and disappear. Lumina which are larger are then expected to inflate and grow over time. And we can also introduce a boundary and ask what happens at the boundary of the domain as opposed to the bulk. So we can consider the heterogeneous case. And just like in classical nucleation theory, we find then, of course, we have two additional interfaces that we have to consider and two additional interfacial tensions. These are called here gamma W1, gamma W2. I will talk about the difference and call it gamma W. This will continue also throughout the next example that I will discuss. And then what we see is that under circumstances where an interface between the droplet and the wall is favored, we get a decrease in the radius of curvature at the same volume. So this is a plot of the radius of curvature as it changes as a function of the surface tension ratio. Below -1, we have a de-wetting regime. So there the droplet would not actually want to adhere to the wall at all. And above 1 is total wetting, so we don't have a stable interface. Instead, the droplet is attempting to maximize its contact area. The reason we became interested in this is because we were approached by experimental collaborators from the lab of Takashi Hiragi. He used to be at EMBL, now he is in the Netherlands at the Ubrecht Institute. And they were studying mouse development, and in particular, they were interested in the role of embryonic and extraembryonic tissue-tissue interfaces. And they had some interesting observations regarding the formation of the lumen in the inside of the epiblast, which is the part of the embryo that later on forms the actual mouse. And this is the lumen here. And we were wondering if we can make some general arguments about the robustness of luminogenesis on the basis of these types of arguments. And so we pose the problem in such a way that we say we are starting from a constant initial volume, from a fixed initial volume, the idea being that basically this framework applies above a certain scale. This is, of course, also true for classical nucleation. We are talking about cases where we already have enough particles that we can actually talk about surface tensions and so on. In this case, probably there are initial processes that set up a configuration somewhere inside the tissue, but we are asking now, once we have a certain starting volume, what are our chances of achieving successful lumen growth, given that we have a boundary or that we are doing it in the bulk. And then we can write this kind of parameter diagram here. Again, the hatched regions correspond to the de-wetting and the total wetting regions. And in the partial wetting region, what we see is that the presence of the wall essentially buys us this yellow part of the diagram. So in the case that the parameters favor an interface between the lumen and the tissue interface, having the tissue interface should promote luminogenesis. And indeed, yes, please.
Sorry, so these lumina are found inside mouse cells? So
they are found inside the tissue, so inside the multicellular aggregate.
Okay.
And they're also found all across biology, right? So the study that I showed in the very beginning was looking at a different stage of mouse development, actually, than the one I'm discussing here. So this is also maybe important to note. But in general, what happens is that the cells, they stick together, but then they can form these fluid-filled cavities in between themselves, and those can become very, very large. And then in the end, you can have like really structures that are mostly lumen with, with a very thin layer of tissue around it. Yeah,
I see. So these are in the spaces between cells.
Yes, exactly.
In the spaces between cells, and
but then in the end you also have like many cells surrounding a lumen, right? So they can become very large.
I see. But so when you draw this axis of the radius, this critical radius and so on. So if I think of classical nucleation theory, but right now you have these surfaces which are your cell walls, and so some radii are not going to be accessible, right? Because of the spatial constraints of the crowding, or is all of this radius accessible?
So the description is in a way very, very effective in the sense that we're not, so the material that is described here is basically just characterized by these tension parameters. Is described as a continuous medium. In reality, it is made up of multiple cells, and there can be few cells or many cells. I mean, I guess in this example that I showed in the beginning, it would be very few cells. But in this study where they look at then the dynamics of the lumina at larger scales, I mean, it's modeled as a continuum, right? So this could be some good number of cells, let's say.
Okay.
And there is no, I mean, we're not taking into account the geometry of vertices between the cells, for example. So this is important, yeah.
Okay, thank you.
Yeah.
I just asked another question while we are at it. So I'm not sure, biologically speaking, so are these lumina eventually evolving into structures, permanent structures like in cysts, or these are just sort of accidental formation of interstitial spaces?
I mean, again, it would depend on the context, what the exact biological function is, but in general, they are considered to be very important for many different things. Sometimes they are transient, and sometimes they develop into structures that are fundamental to organs. I mean, like many of our organs have are basically epithelia that surrounds large volumes of fluid.
Right.
And
so when,
but they can also be transient. I will actually, this will not be the focus of the next example, but it features also a lumen that is doing interesting things and eventually disappears, let's say. Something else that I think in also more recent years is gaining more attention is lumina as signaling hubs, meaning that there are molecules that are being secreted into these cavities and then they diffuse and they can communicate, basically mediate communication between other cells, which I think is very interesting because then also you have some coupling between the biophysics of these objects and the signaling interactions that can go on.
Okay, thanks.
Yeah, so basically just to very quickly finish this part, what our collaborators found, and they were mainly interested in finding a way to grow these embryos outside of the uterus so that they could take they could do live imaging, meaning they could take movies and get a sense of the dynamics of the development. And they had two different conditions that were working for this. In one case, the embryonic extraembryonic interface form in this way, and this has this flat interface. Basically, what is necessary for that is that the extraembryonic ectoderm here undergoes a morphogenesis and folds inwards. And they had a different condition in which this was not taking place. And in that case, this flat tissue interface was absent. And so we could compare basically the success rate of lumen expansion in these two conditions, and indeed found that in the presence of the embryonic extraembryonic interface, the lumina formed more robustly. The other, in a way, prediction that comes out of this is that the lumina indeed should be positioned at the interface as they grow. And this also is something that was not easy to measure, because all you can do if you don't have live imaging is just take some snapshots. And here also what we found is that because they had gotten the live imaging to work, the vast majority of cases, the lumina were actually positioned at this tissue-tissue interface, which is not how it's drawn in most of the textbooks, interestingly. And this is work that we are also now following up with a postdoc, where we're now looking also a bit more at the interactions of the cells and the role of cellular polarity in this process. But for this talk, I will stop here and basically just summarize that in the end, what we use here is very simple equilibrium arguments, but one can maybe draw some conclusions from that about developmental robustness. In the next example I will discuss, I stay actually with a case where we can remain with descriptions with constant surface tensions, but we will have non-equilibrium movement of contact lines as a consequence of an external broken symmetry in the confining geometry. And we are also staying with the context of mouse embryo for this. Before going into it, I want to just spend one slide emphasizing also that these types of concepts, so wetting theory, description of things as objects with surface tension, is of course also very widespread in the context of cells interacting with substrates and also multicellular aggregates interacting with substrates. And I want to highlight just a few recent papers that were doing this. And also, I think in the physics community, there is more interest now in interesting wetting phenomena, soft adaptive wetting and elastocapillarity and things like that. And I would say that these biological systems provide very nice systems for testing, experimental testing of some of these ideas. And the interface, the interfacial interaction that we were studying, this again was collaboration with Takashi, the grad student who did the experiments was Vlad Bondarenko, and Roman from my group did the calculations and the simulations. And we were looking at the mechanics of maybe the most important interface that we have in our mammalian layers, namely the one between the embryo and the uterine wall during implantation. And essentially what happens there is that the cell layer at the outside of the embryo, the trophectoderm cells, they engage in a very complicated interaction with the uterine wall in such a way that over time the embryo becomes completely embedded inside the endometrium. And in vivo, so from the actual uterine environment, one can only do fixed images and fixed staining. So we get these snapshots from different developmental time points. Here, the blue dots are the nuclei, both of the uterine wall. So mostly, basically, this part here is the endometrium, and then the embryo itself, their actin is labeled in green. So this is the embryo, and what we can see is that this is a very complicated process. The trophectoderm cells, they form these large protrusions. They seem to really invade into the uterine wall, which is soft and deformable. There are complex biochemical interactions going on here, and from a physical point of view also, this looks like a very complicated problem. Our collaborators, again, were interested in obtaining a platform where they could do live imaging, so they could quantitatively measure the shape changes and other processes inside the embryo over time. And we were very happy that they teamed up with the lab of Matthias Luttorf and approached, relied on an engineering approach, where the idea was that they would create an artificial environment which mimics the uterine wall, and then they can put the embryos inside and make them grow. And they optimized the geometry quite a bit, but then they ended up with these frustum-shaped PDMS mold-based hydrogel wells and managed to actually get the mouse embryos to grow inside these wells and to develop properly until a certain point, at least. So I'm very casually summarizing, I think, very, very extensive and hard work here. For us as physicists, it was very nice because this, of course, allows us to study some aspects of the mechanics here in isolation. So instead of dealing with a complex soft wetting problem with bidirectional signaling interactions, we can here consider the geometry of the confinement to be fixed, and we can focus on describing the shape changes of the embryo inside this confinement and hope to learn something about the mechanical changes that happen during implantation. And essentially the hypothesis we had is that the embryo is relying on a kind of capillarity effect in order to insert itself into these wells. And we wanted to start with a very, very simple description that allows us to capture the essential aspect of this. So we model the embryo as a simple droplet with uniform properties throughout, confined in this geometry, which we take to be fixed. And then we have basically, again, three different interfacial tensions. Gamma W again denotes the difference between the medium wall and the droplet wall tensions. And then gamma naught is the tension between the droplet and the medium. And we can go to an even simpler physical limit. We can take the confinement to be cylindrical. In that case, we get analytical solutions for the shape of the droplet. Here it is parameterized in terms of the heights of the caps. Again, we get out the partial wetting regime. Below -1, we have total wetting. At some point, we should get to topological transition in the middle where the two sides fuse. Above +1, we get de-wetting, and in the middle, we have this branch of solutions here. And then qualitatively, the idea is that if the embryo, this was the hypothesis, and this is what also much of the biology literature suggests, what happens is that the trophectoderm cells on the outside of the embryo are increasing the adhesion to the uterine wall. So there's a change in the expression and surface localization of integrins molecules that mediate binding to the outside over time. And then that essentially would correspond to movement of the system along the branch in this direction, because we would have a lowering of gamma W as a consequence of this. And indeed, qualitatively, when we compare images of the embryo taken at different times, we see a characteristic change in the contact angles that agrees with qualitatively with this type of change. The other thing we can say immediately here is that if we perturb adhesion, if we prevent adhesion, we should see that we enter the de-wetting regime. And indeed, there were experimental conditions in which the wells of the confinement were prepared in such a way that they disrupt the interaction with the cells. And in that case, we see these characteristic maximum contact angles when the volume is above the confinement limit. What also happens in this system, because the volume is interesting, it is very dynamic as a consequence of the lumen that is inside the embryo. It's a different one. This black space here, that's actually a fluid filled cavity that undergoes very interesting inflation collapse dynamics, the physics of which has also been studied and is very interesting. But here, all I want to say is that sometimes the volume does go below the confinement limit. And then when we are in this de-wetting part of the diagram, we indeed see the embryo detach from the wall and take on a nearly spherical shape. So we wanted to also make quantitative predictions for the shape dynamics and actually infer from that more precisely how the adhesion would change over the course of implantation. And in order to do that, we now take into account the broken symmetry in the confinement. So we put this frustum-shaped geometry. Then we are no longer translationally invariant, and we have 4 degrees of freedom that describe the shape of the embryo. We are assuming that the free surfaces relax to near spherical shapes. So the idea is that that changes faster than the contact lines. And then we have the 4 degrees of freedom, which are the heights of the caps and the positions of the 2 contact lines. We also relax the assumption of equilibrium. So we are writing dynamical equations for these 4 degrees of freedom, introducing these general dissipative coefficients here. And then there's basically 2 processes that do work, that drive these dynamics. The first one comes from the tensions. This is also where the change in adhesion enters. And the second one describes the effects of the volume dynamics that I just explained. And what we do here, because we don't explicitly model all those complicated processes that result in changes in the volume, we actually measure the volume individually for each embryo, and then we put that into the equation directly from the measurements, so that we can learn something about the contribution of the adhesion changes. And Roman was doing simulation-based inference here to estimate the parameters that govern the decay in adhesion, essentially. And so we do that individually for each embryo using the individual volume dynamics. Here are 3 cases showing the images of the embryos together with simulations. Let me know if there are any issues at any point with the videos. And we can fit the contact angles quite nicely. We also get quite consistent values out of the different embryos, even though they look quite different. We also did some validations since then to confirm everything. And basically what we can learn from this is pinpointing the window of time and the extent to which adhesion between the embryo and the substrate changes. Of course, now it would be really wonderful to have measurements also of surface integrin levels or something like that over time. At the moment, we don't have that available. Instead, what we can do to at least get some sense of whether this is in the right ballpark is to take as a proxy hallmarks of a morphogenetic process that is known to occur at the same time that the tension release is happening in the trophectoderm, and look at the shape changes that accompany this morphogenetic process. And what we get out is still quite nicely consistent in terms of the window of time. Now, we can, of course, generally also characterize a little bit how this geometry is driving the actual motility. So how basically in terms of the motility mechanism. So we can take average values of the parameters and the volume and then see how the velocity of the contact line changes as a function of the frustum angle. So the experiments live here. We have a maximum. And then as we go to 1, that's the geometry where we essentially are dealing with a flat substrate. And that can also be done experimentally. When the embryo is put on a flat substrate, it spreads out consistently with this wetting idea that we have. But what happens is that the embryo gets crushed, so it cannot develop properly. And in the end, what we think is important here is that the contact line moves with a velocity that matches an internal biological growth timescale that describes the growth of the embryo on the inside, so that the embryo actually has enough space to grow as it develops. So again, just another example of what it looks like. You see the inflation collapse of the lumen here quite nicely as well. The pink stuff is what will later turn into the embryo. And because of the confining geometry, it has the space to elongate and to acquire the structures that it needs. Of course, in vivo, the whole thing is solved in a much more elegant way, because there the embryo self-organizes its own geometry. We should more realistically think of the embryo as an active droplet that embeds itself into a soft substrate. And then it can deform the uterine wall and acquire exactly the confinement shape that it needs. Physically, it would be more complicated to study, but not impossible. And hopefully also at some point, imaging data will become available of implantation in vivo. So in this case, the motility overall was driven by the external broken symmetry in the geometry, but many cells and many multicellular aggregates as well, as I said in the beginning, are capable of generating gradients in components of the tension. And then these intrinsic stress gradients are able to do work on the contact line and lead to self-propelled motion. And so I just want to show one very simple example to kind of explain the principles. This is from a very rather old theoretical paper, but they show a very nice derivation. We imagine a droplet that is sitting on a substrate. We can parameterize a linear gradient in the tension which is decreasing in the direction of movement. And basically, we obtain an expression for the velocity by considering the balance of forces. So we have a driving force that is proportional to the magnitude of the gradient, delta gamma, and then we have a drag force that is proportional to the velocity with the drag coefficient lambda. Here, sorry about the symbols, it's not a, before it was a dissipative coefficient. Here it describes the size of the droplet. And actually, this study was not motivated by looking at cells. It was motivated by systems in which, so one can prepare such things also for passive droplets by taking substrates that are coated with chemicals in such a way that one gets a gradient in the adhesion. And then you can get motion also of passive droplets. Similar things can be done with temperature. And I really like this video. In this case, the authors put droplets into mazes that are prepared with chemical gradients inside, and you see that the pink droplet is successfully finding its friend over here, even though it's a completely passive system. But of course, cells do it themselves, so they can themselves intrinsically regulate such gradients. Here, again, relatively old movie of a cell crawling on a substrate, and the adhesion complexes are labeled in green. You see that no matter how it turns, the gradient is always scales to the size of the cell and points in the direction in which the cell is moving. Now, for crawling cells, for migrating cells, this kind of stuff has been studied quite extensively, both on the theoretical and on the experimental side. And so for the next part of my talk, I would like to talk about a slightly different situation in which we actually have two droplets with gradients coupled to each other with opposite gradients. And then we will see what happens. It's already, I give it away right away. So what we see then is the work that these gradients do results in the rotation of the contact line. And this is work that I did while I was still a postdoc in New York. And we were inspired by observations from zebrafish sensory organ development, in which there are cells that come in pairs, and they form this characteristic soap bubble doublet kind of configuration that you can see here. And we had reason to assume that these cells over time acquire a polarity that introduces gradients in the tension at the outer surface. So this surface probably gradients in adhesion at this surface with the cells that are on the outside. So these cells are surrounded by other cells. And we also knew that the cells engage in a mutually inhibitory signaling interaction across their interface. And these tension gradients are always oriented along the anatomical axis of the organ and the animal, the fish in this case, the anteroposterior axis of the fish. But the sign of the gradient, so whether it points in the positive direction or the negative direction, is set by this mutually inhibitory interaction, which we thought was unbiased in the sense that the doublet does not know about its own orientation relative to the organ or the fish axis. Rather, it is just a matter of tug of war. One of them wins, and 50-50 which one, so that we end up with 50% in each possible configuration. So we can have doublets with gradients that point towards each other and doublets with gradients that point away from each other. We can call them positive and negative dipoles. And we can use the exact same arguments that I showed before. We can essentially look at the balance of forces at each point on the contact line. We can parameterize it here with RC. We can calculate the torque that acts on the contact line. And again, from the conservation arguments, we get that the angular velocity of the contact line should be proportional to the magnitude of the gradient. And so we can write it as a dynamical equation, actually, of the angle of the dipole of the doublet. And then we see that we have an unstable configuration in the negative dipolar configuration and the stable configuration when we have the positive dipole, meaning that any small change around this configuration should lead to the rotation of the interface and the system transitioning into this stable point here. We get the exact same result if we simply write down the free energy of the surface with the surface tensions that vary along the organ axis in this way. And then we recover that this surface potential has a cosine shape with maxima at the negative dipole configurations and minima at the positive dipole configurations. in the experiments, we indeed observe rotations of the interface, very, very precise rotations that always go 180 degrees. Sometimes when the cells are initially oriented off angle, then they rotate exactly the right amount so that in the end they are aligned with the organ axis. So here I'm showing a video, and there's also some panels in case the video is not so reliable. Whoops, sorry, I will show it again. So you see the interface rotate at the center, and Adrian did a lot of live imaging of these doublets and recovering what we thought initially, that the signaling interaction was unbiased and produced 50-50 of each configuration. We saw that pretty much exactly half of the cells had a rotating interface, the other half of the cells had no rotating interface, and also the actual angular velocity matches with what is predicted here, with a theory that is taking the gradients just to linear order. So it probably simplifies the picture quite a bit, but we get very nice quantitative agreement with the experimental measurements. Here we are fitting just a single parameter. So this is just one thing that we're adjusting here. And what we can also do is we can look at the shapes of these doublets. So of course, the gradients and the tension introduce deviations from the constant. So when we have constant tension, we have constant mean curvature shapes. This changes when we have a gradient. We can calculate them to linear order in the gradients analytically, actually, for the axisymmetric configurations that we have in the two dipolar cases. So for the positive dipole and the negative dipole, and what we get out, again, the expression depends only on the ratio of tensions and the contact angle, which we can also self-consistently get out of this framework. I'm not discussing that today. Basically, what we get is that the negative dipole should be kind of squished a bit like this, and the positive dipole is elongated like that. We see that also in the data, and we are able to fit the shapes very nicely as well for these two configurations. Yes.
Sorry, can I ask a quick question? So this doublet formation, this you are looking at what stage of development in zebrafish?
Yes, so I have the final slide is a bit more about the biology context, right? So what happens is that, actually, let me see, maybe I just advance one slide and then it's easier to discuss. Okay. So of course, why do the cells do this and what's the point? Basically, these are part of the lateral line organ of the zebrafish, which is their primary sensory organ. I think it is safe to say that. So fish by far probably rely the most on sensing water currents in their environment. And so these are small patches of mechanosensory epithelia that are distributed along the side of the fish. And each of these organs, as it develops, has these sensory cells inside. And each sensory cell has a mechanosensory organelle that responds to mechanical deflection. And these mechanical and the mechanosensitivity, so the sensitivity of the sensory response depends on the direction of the stimulus relative to the orientation of the organelle. So what I didn't mention is that the polarity that I just talked about that sets the sign of the gradient along the axis is later on governing also the orientation of the mechanosensory organelle, meaning it determines the sensitivity of the cell to mechanical stimuli. And so what we think is going on here is that the fish in this way is able to produce these bilaterally symmetric organs that have the same number of positively oriented and negatively oriented cells. These are innervated also separately, very interesting process also how that works. And then we end up with an organ that has an architecture that favors sensitivity of stimuli that are delivered along the AP axis of the fish, which is probably, for an organism that is swimming in this direction, it's a very important direction, let's put it that way. So yeah, if they don't want to be eaten by sharks and so on, it's good to have a very robust mechanism of generating these lateral line organs that are looking like this. Now, developmental stage wise, this is at the larval stage where this development happens, but these organs also have a really incredible regenerative capacity. And the exact same processes happen during organ regeneration, also at later stages. And in fact, the data in the paper are taken, so we studied both regeneration and development.
Okay, thanks.
And they come, so these cells, they come in pairs and they don't all come at the same time. So that is also interesting. You have 2, 4, and so on. And later there's also some homeostasis where they keep getting replaced. But always you have these pairwise interactions. And I find this also quite interesting, how in the end, these are pairwise interactions, biochemical interactions, and then these mechanical processes, and they give rise at the scale of the organ to this bilateral architecture that is clearly relevant also for the survival of the organism.
Okay.
Yeah. So then I will switch gears a little bit for the last part. I will continue talking about multicellular patterning processes, but from the point of physics, I would say that this project here was a little bit more about how actually gradients in tension can arise in a biological system. So far I have discussed, assuming there is a gradient, what happens in terms of the work on the contact lines and the shapes and so on. But it is also interesting to ask how to actually pattern a biological surface and how to get such gradients out. And so in this case, that's a little bit what I'm going to touch upon. This also is a collaboration that I took from Rockefeller with the Laboratory of Morphogenesis there. And the motivation came from understanding the principles underlying patterning in the skin, in particular, the spacing of feather follicles in the avian skin. And so these are images from the skin of chicken. And over time, there is this beautiful process where mesenchymal cells aggregate into first a streak, then a set of aggregates, and then this sort of more or less hexagonally distributed configuration here. And our collaborators had, in my opinion, a really, really ingenious ex vivo way of studying the patterning principle. What they do is they take the dermal cells, the fibroblasts, from the skin of the fish, they suspend it in a drop, and they put this drop on a collagenated surface. And then the cells by themselves settle at the rim of the droplet so that we have a ring at the end. This is probably a coffee ring effect or something like that. And then on these rings, we can observe spontaneous pattern formation. So I hope the video plays. If not, there is a time sequence at the bottom. The cells, they move around randomly, and then they aggregate and they form these equally spaced clusters. I was, of course, infatuated with this. I mean, patterning in 1D and periodic boundary conditions, it's perfect. But also, the other thing that I found very interesting here is that the cells engage in a very interesting interaction with this collagenated substrate. So from the top, it is not so easy to appreciate, but from side views, we can see that these fibroblasts, they really insert themselves into the collagen. They exert forces. They also secrete collagen themselves, and they introduce irreversible rearrangements in this extracellular matrix material. And so we can view this as an active contractile viscoelastic material. And on the time scale of patterning that is relevant for understanding this process, tens of hours, we can consider this to be a viscous system. And then we can write down very simple active hydrodynamic equations for this one-dimensional system. The first equation here is, again, basically a statement of momentum conservation, force balance. So this is the equation for the velocity. We have friction with the substrate and viscosities, which gives us the hydrodynamic length that governs the range of flows. And then the flows are driven by an active stress that is, in this case, taken very simple, proportional to the density of cells that are thought to exert active contractile stresses on their environment. They act as active contractile dipoles. The second equation describes the evolution of the density of cells. It's a convection diffusion equation. First term is transport by convection, and the second term is the diffusion term. We can estimate the diffusion coefficient very nicely from the experiments because we can track the random movements of the cells. And this description is completely analogous. So it's basically the same description that we have for active fluids, also at different scales. And it was motivated initially by looking at actomyosin, so motor proteins inside the cytoskeletal filament, resembling this, but at a very different scale. I will come back to that at the end, because in and of itself, I think this is interesting. And these types of active fluids, they can undergo spontaneous patterning. So in the paper, we call it normalized contractility. This is basically the Peckley number, meaning it's the ratio between convective and diffusive timescales. When the Peckley number is larger than 1, meaning convection wins over diffusion, we get spontaneous formation of aggregates. The timescales vary quite dramatically. So here I highlight the 50-hour timescale just because below that, I mean, above that is where it becomes close to being relevant for the actual biological process and the experiments. And then using linear stability analysis, we can immediately make predictions about how the number of aggregates, the wavelength of the pattern should change as a function of the active contractility. on the one hand and the hydrodynamic length on the other hand. And in this ex vivo system, we can perturb these quantities using different drug treatments. So these are different drugs that modulate, upregulate, and downregulate myosin contractility inside the cells. And the viscosity of the material was modulated by glycosylation of the fibers. We cannot make really fits here because we don't know how to convert these drug concentrations into actual magnitudes of stress perturbation. But I want to emphasize there is only one parameter that we don't know. Pretty much everything else is constrained. The parameter that we don't know is how the density of cells, the factor of proportionality between the density of cells and the stress. And just with this, we get quite reasonable quantitative agreement in terms of the number of aggregates, I would say, relatively considering the changes. The other thing, we can of course also do simulations, then we get out what the patterns look like and how it evolves from it. So we have to do a small noise perturbation in the beginning because the perfectly uniform state is, I mean, it's unstable, but it would stay like that. And then what we can also do, and now without adjusting any parameters, is to look at how the time scale of patterning changes as we do these perturbations. So here I am showing the 3 glycation experiments, and then also across all of the contractility experiments, we get really nice agreement, I would say, in terms of the characteristic time scale of pattern formation. One thing we see in the simulations that we don't see in the experiments is coarsening. This is a very generic feature also of these active fluid instabilities. So over time, the linear stability analysis gives us the first dominating mode, but over time, these aggregates the peaks are expected to fuse. And in the end, we should end up with just a single large peak. This does not happen in the experiments. We think that that's because eventually these aggregates, they deplete the material in between themselves so that we stop having an actual continuous flow system. And also in vivo, there are processes that lock in the initial patterns. There is signaling that reads out these initial changes in the density. I would say it's important to think about on what time scale do these patterns matter in biology, and then sometimes doing linear stability analysis is all that one needs in order to understand what happens. And yeah, basically just to finish, I want to emphasize again, what I found interesting here is that it appears that the same physical principles govern pattern formation at very different spatial scales and also or levels of biological organization. So I want to come back to the fact that these active fluid instabilities apply to cytoskeletal networks inside cells with myosin, where the contractile elements are not big fibroblasts, but are the myosin motors that are inside those cytoskeletal networks, and we get same types of patterning at this very different scale. Here is an image of a cytokinetic ring on which we get cluster formation of myosin motors. And yeah, with that, I'm done. So I would like to thank you all, of course, for your attention. I also want to thank the group and my wonderful collaborators. So these were all collaborative projects. I have not presented, I have barely presented any of the work that is ongoing. So please stay tuned, and I'm also happy to discuss. Yeah, and thank you. I'm happy to take any questions if there are any remaining.
Thanks a lot, Anna. That was a wonderful talk. So if people have questions, please raise your hands and then we can take.
Otherwise, I might use this opportunity to do a bit of shameless advertisement.
Please go
the
ahead.
MPhD program is open at the moment, and it looks very much like a biology program, but there are a number of us that are really, really keen on having physics students. So, you know, if any of you in the audience are interested, don't hesitate to reach out and talk to me about that.
Yeah. Hi, Anna. Thanks for the very nice talk. So I have a quick question regarding the last part of your talk. So where you show these patterns and introduce the hydrodynamic model. So so here they put the cells like on a collagen on a circle, like on a system with on a substrate with polar symmetry, right? So sorry.
No. So there's no micro patterning of the substrate. This is one of the things that is also very nice about the setup. So it's just a collagen layer everywhere. And then they put the cells in a droplet, they pipette the droplet onto the substrate. And then the cells, we didn't study the physics of how that happens, but the cells, the suspended cells, they go to the surface of the droplet and then they form a ring by themselves.
Okay. No, so I was wondering if instead you would have taken like a square substrate or anything else, like if you would see any kind of like 2D pattern, or there are scenarios where you can also see something where you see patterns in 2D.
Yeah. I mean, in the chicken skin, of course, it is 2D, and one can, of course, also describe it in 2D. They wanted to have a 1D system, and I think they tried to actually make lines, but making lines is harder than making rings. And you would then also have boundary effects, right? So it's with the periodic system, it's an advantage in the sense that it really lends itself to the kind of
Right. And just just to be clear, so in the model, so each of your cell is a contractile element, right?
Yes, we're not modeling them all, right? So it's described again as it's described as an active fluid.
Yeah.
Yes.
Okay.
With a density field and a stress field that is proportional to the density field.
Right.
Yeah, but the assumption is indeed that each fibroblast will contribute an active contractile stress. There is a lot of work, of course, on fibroblasts being contractile in terms of the force distribution that they exert on their environment. So this was, I was comfortable making that assumption. But,
right. I think there is a question by Chaitanya. Hi, Chaitanya, please go ahead.
Wonderful talk, Anna. I'm sorry, I may have missed some things in the beginning, but in the last part, when you made a comparison of the multicellular system to something like an actomyosin system, do you think, since your sort of phase diagram of patterning has length and I think some normalized contractility in it, would the numbers change, but qualitatively things will remain the same? And because I would imagine everything scales down to smaller, so would everything just shift left effectively, or how do you imagine this will happen?
Yeah, that's an interesting question. I mean, for example, the hydrodynamic length, right? That's a physical length. We did not measure it directly, but there were numbers reported for other like large scale systems, which are also driven. I mean, what's also interesting is that the actual contractile mechanism is actomyosin still, right? So what drives the contractility of the fibroblasts is actomyosin also, but it's a distribution of and also interaction with adhesion, of course. Maybe it's a bit more complicated, but still probably the actual the fuel consuming elements in both cases are myosin motor proteins. And we had an estimation of the hydrodynamic length being on the order of 100 micrometers, also from other works that have studied sort of large scale supracellular actomyosin flows in zebrafish embryos, for example. And this was also a value that for us came out as reasonable based on semi-quantitatively, let's say, match the change in the number of aggregates with the magnitude of the drug. So in fact, what you can do, and it's very silly, but what you can do is here. So we have these different treatments, and what you can do is just make a very, very stupid assumption of the different drug perturbations, introducing linear changes in the stress equally spaced, and you can fit this curve very nicely, very nicely with the hydrodynamic length that comes out to be about 100. So that's how we did it in the end. Of course, inside the cell, I have never thought about it sort of systematically how these scalings would work, because each individual myosin mini filament is a few hundred nanometers, right, or smaller, and though the range of flows correspondingly is, again, I don't have the numbers off the top of my head, but it's
fine.
Somewhere on the scale of a few microns. So there is scaling there. yeah, this is interesting, because then one could of course ask, or what are the constraints between, can we pattern faster on smaller scales than on larger scales, and these types of things. Yeah, very nice.
No, thank you.
If I may, I had one. Sorry.
Yeah, go ahead.
No, sir, I had one question. So what controls the width of the ring that you see? So if you pipette, like in the droplet, if you put more cells, does that increase the width, or does it just increase the density of this boundary layer of cells that you have? And does that actually change the pattern formation?
Yeah, this is a good question. So I'm not the experimentalist, right? So I also don't want to say anything wrong. So they experimented with different ring sizes, and you can change the concentration of cells, for example, in the droplet, and then that will change from what I remember, what that actually changes is the width of the ring. So it looks like there is a more or less fixed surface concentration. Surface density, so which also makes sense, because in the end the contractility comes from the fibers interacting with the ECM. They do embed, so microscopically again it's not 1D at all, but there is a limit. I mean, they can probably there's a packing fraction above which they cannot act as this kind of contractile elements anymore, and then more larger concentration, if I remember correctly, changes the width of the ring. Now, yeah, so that would, what that affects is the initial. So if we then project everything onto a line, we have a parameter which is rho 0, which is the initial, the concentration of the initial steady state, which is unstable, and it does affect things.
Nice.
So it plays a role. Yeah, and we took it from the experiments for the simulations, but yeah.
Thank you. So sorry, Amitav, I think you were asking something.
No, so I think I still had a doubt. Like, can you go to the next slide? So if I just wanted to understand if I misunderstood. So this contractility, when you say coarse grain the whole picture and writing down a continuum equation, so this contractility is actually not at the level of individual cells per se. It is like as if like two cells actually try to, or few cells try to come close to each other. That is like something I should understand as a contractility. Like in actomyosin, I can understand like why it is contractile. So here, so what is the origin of the contractile stress locally, if I try to understand?
Yeah. So it's the whole layer is described as a continuous active fluid. That's the point, which has a density field of contractile particles inside, and this spatially non-homogeneous active contractile stress. And then we write a constitutive relation for this active stress, and one can also use different expressions there. So for example, in this paper, which was motivated by actomyosin, they use a saturating function, which is also sensible because, of course, above a certain density. Because we anyway wanted to essentially focus on the initial stages of patterning, we just said we are going to keep it simple and we make it linear. But that's essentially what it means. Now, if your question is more kind of like mechanistically, how do I generate large-scale active stress with individual contractile cells? I think the best way to think about this is to again think about, so in the context of actomyosin, by the way, this question is also not completely answered, or at least it has motivated a lot of studies, right, where people try to understand at the microscopic level, why do we see contractility come out? And there are also different arguments that people have made. I mean, some studies do simulations of filaments and the motors, and then you can find arguments that say that the contractile configurations of myosins interacting with filaments are more stable and more frequent, and therefore, so there's arguments like that. So, and one would essentially have to extrapolate those types of arguments to a system like this. My motivation for going with this is that in the end, what we can, I would say we have a big body of theoretical and experimental literature that shows us that fibroblasts exert the force distribution that they exert on their environment. The dominating mode of this is positive contractile dipole moment.
Okay.
Therefore, we can consider fibroblasts as positive as contractile dipoles. Then if we have a density of contractile dipoles in some large volume, this should give rise to large-scale contractile stress. In fact, I would say in this case, it's experimentally more substantiated because you can really do these experiments. You can put a fibroblast inside collagen and measure how it starts contracting everything around itself. And yeah, maybe I could have found actually a number for the scaling coefficient somewhere in the literature if I would have looked hard enough. Yeah.
Okay, thanks. So are there any more questions? If that is not the case, let us thank Anna for this wonderful talk once again.
Thank you. It was a lot of fun. Thank you very much. Yeah.
Thank you. Thank you.
So I will end the stream.
Yeah.
And